Welcome, everyone. My name is Adam Kaufman. I'm a board member at the CMTRF since 2019. I'm also a patient. It's my pleasure to introduce Robert Pryor, a postdoctoral fellow from VIB-KU in Leuven, Belgium. He'll be talking about drugs under development for CMT. Welcome, Rob.
Okay, I'd like to say thanks to the CMTRF for inviting me again to give this little update. I will say this is a talk tailored more towards CMT patients and not towards researchers. And a bit of the talk is just being covered by Mary. So first of all, who am I? I'm a CMT X-linked type 1 patient myself. And as you may be able to tell from the faint Irish accent, I'm originally from sunny Ireland. But growing up in Ireland after a diagnosis of CMT, there was actually no real information for myself or my family, and there was no CMT patient organization in Ireland. In addition, there was no CMT research in Ireland. I was super interested, me and my family were super interested in actually the research and what could be done, but there was nothing for us. And so then growing up, CMT really represented this like unknown, this uncertainty for a CMT patient. You don't know what's down the road for you. And for the CMT patients in my family, there was a huge variation between family members. I grew up with two cousins. We were all the same age, and it varied from mild, moderate to severe phenotypes from the age of 11. And for my mother, she was also in a wheelchair, and she's, as you may have heard from Mary Riley's talk just beforehand, the CMT1X or CMT X-linked type 1 patient, female patients can vary quite a bit in their phenotypes. And for my mother, it was quite severe. So CMT represents uncertainty. So I decided to become a CMT researcher and pretty much dedicate my whole career to CMT research. And when I was looking to move to where the action is, there was a paper that was published from Belgium. Belgium actually has a strong history of CMT research. The first gene was identified uh, in Belgium by Belgian researchers. But I moved to this young man's lab, Professor Ludo van den Bos, and Ludo and his group had just published a huge paper in our field, and it showed the use of histone deacylase 6 inhibitors could reverse the CMT phenotype or the disease characteristics of a mouse model that they developed. So it can completely reverse the phenotype. And this was super interesting. So when I joined there, I got to be a part of the research, and I worked on many other things, but including the HDAC inhibitors. And during my PhD, we got to actually launch a company based on novel histone deacylase 6 inhibitors. And this company is Augustine Therapeutics, and they're now a clinical stage biotech company that are translating HX6 inhibitors for the treatment of CMT. That was super exciting. That was quite early on. I got to achieve part of my life ambitions. So that's nice. But enough about me and more about CMT research. As you heard from Mary and the dramatic introduction video at the start about there was very little done in CMT research for the greater part of the 20th century. All that could be done was clinical observations. So a patient comes in and their disease characteristics are monitored and reported, but nothing was really known about the genetics behind it. And until there was an inflection point, what I would say, and that's really what Mary also pointed out, was the discovery of the duplication of the peripheral myelin protein 22 gene that causes CMT1A. And since then, there's been over 130 genes that have been shown to be disease causing in CMT. This has been enabled a lot of pathways to be discovered because of that. If you know the genetics, you can understand what pathways are involved. And I will say I was born in 1991, but it has nothing to do with these academic publications. So I hear what you're saying, all these academic publications, all these genes, but where are my drugs, Rob? So where are all the therapies, all this knowledge, all these genes? Um, it's actually very difficult to develop therapy, especially with a slowly progressing disorder. So, like I said, for the best part of the 20th century, it was clinical observations for patients and just reporting of their disease symptoms. And then once we found the genetics, and I'll just point out a handful of papers, once we developed the genes associated with CMT, you can really build upon this. And as you can see, some of the papers here, the identification of the peripheral myelin protein 22 gene, and mitofusin 2 for CMT type 2A, and several others, especially with the connexin 32 or the GJB1 gene, which I have in my family. So a lot of these papers were huge multi-centered studies. So it's understand that it's not just one small group, typically not, that develops these. It requires a huge collaboration across different research groups to identify the genes. And once we have these genes, we're able to understand the cellular pathways. And I'm just going to mention one or two, including my own. We're able to understand the role of PMP22 and how it's important in lipid metabolism and homeostasis in Schwann cells, and as well as axonal transport being associated with CMT type 2. This allows us to then build upon and develop therapies for these cellular pathways and for CMT. But a massive issue, which Mary touched upon also was, oh, going back, was the clinical outcome measures. How can you measure sensitively enough a slowly progressing disease? Because where this is an issue is if it takes too long to measure a small change in the disease, it means that the clinical trials need to run for a lot longer, which means it's a much more expensive investment for companies, and companies don't want to invest a lot of their money. It's going to cost hundreds of millions to conduct these trials for something they're not sure that's progressing fast enough to measure a significant difference. So this has been a huge issue. And what I will mention is that obviously Mary's been pioneering this field, clinical outcome measures, and lots of other groups. That's really to help advance the clinical trials. We have the therapies. We need to know that these clinical outcome measures are sensitive enough to move forward. Another important element is natural history studies, and this is very important for rare diseases because if there's not a lot of patients, um, for a clinical trial study, natural history studies can be used as a reference point to look at the disease progression. And therefore, the patients with these ultra rare diseases can actually be all treated and be compared to the natural history. So, as I just mentioned, why rare disease natural history studies are important. You get to map out the disease progression over time in patients, going from early to adolescence, and then you get to measure really where the significant declines are in these patients throughout their lives. And that enables you to design clinical trials better, as well as when to treat these patients. So if you know when the disease significantly worsens, to treat before that pre-symptomatic. That's the idea of these studies, and it's very important that patients do get involved in these. Okay, what's in the therapeutic pipeline for CMT? As mentioned earlier, we had the ascorbic acid trials early on, and this is just a graph looking at the number of clinical trials over the past 35 or should be 33, well, yeah, 35 years. Thinking about my own age, sorry. So as Mary pointed out, the ascorbic acid trial, and there were some other smaller trials, including the coenzyme Q10 trials and Farnex trials, and these are huge learning opportunities about the sensitivity of these outcome measures and where do we go from here, and if we can measure the disease progression uh, sensitive enough. From 2019, there was a bit of a pivotal change, which I would say is a new inflection point for CMT research and drug development. There's a lot of new therapies that are coming in, a bit more sensitive and targeted. We have a lot of gene therapies, cellular therapies, and very specific pathway therapies um, that I think will continue to increase the number of clinical trials that we see in the CMT area. So again, what happened between 2019 and 2025? Well, I tried to map out the companies in 2019. I will mention this may reflect survivorship bias. So I'm not going to mention too many of the companies that had failed, but I did leave in Farnex, which we know that the company did not have positive phase three results, and they dropped out afterwards. But there's been a huge increase in companies. We have roughly 37 companies registered that are translating therapies for CMT. And I do want to mention actually the role that patient organizations can play in accelerating the advancement of these therapies. And the CMT or I've had a very successful program or funding campaign, the CMT 1A Endgame. This did actually result in almost 30% of the companies you see here being funded. And that means you're really enabling companies to develop their CMT programs. Some of them may not have been on CMT, and the CMTRF say, "Hey, come work on CMT. We'll show you how." So, CMT or patient organizations can really partner with companies to help advance their programs and help design the programs better. So yeah, between 2019 and 2025, it's almost a 3.x or 3.6x increase in companies registered with CMT programs. I will say this does not reflect universities. There's a huge amount of research being done on CMT, and a lot of the early gene therapy uh, studies are actually being done in universities and get eventually translated to companies. So, what are the therapeutic classes under investigation? Again, Mary kind of covered this, but it's small molecules, and this actually represents 55% of the current therapeutic treatments that are being advanced for CMT. These small molecule compounds typically target enzymes or pathways or organelles in the cell, and they help improve either viability, energy, metabolism in the neurons or Schwann cells, or help improve the cell-to-cell connection, whether that be the neuromuscular junction between the motor neurons and the muscle. Then we have gene therapy. As we heard, this can cover a variety of different techniques, but typically you're going to have a gene modifying therapy introduced into the affected cell type, whether that's the Schwann cell or the peripheral neurons. And the concept is either to introduce a corrected gene or to modify the disease altered gene. Uh, and a big hurdle for this field, for this area, is actually the delivery mechanism. And so there's been plenty of different techniques being investigated, whether that's viral vectors or lipid vectors or vesicles. But delivery is key for this field and for the whole area in general. And gene therapy, I should say, represents about 35% of the current uh, therapeutic interventions being looked into for CMT. Transplantation is another one, and that's where you can have either autologous or allogenic cell transplantation. That's either where it comes from the patient themselves, they have cells isolated, turned into stem cells, and turned into the diseased cell type, or well, the corrected cell type, hopefully. And that would be, say, for instance, Schwann cells for CMT type 1. They can also be sourced from other individuals, healthy donors, and but then that can also cause certain issues. I'm not going to talk too much about that. And then there's another class I would call, which would be looking to modulate different cells, and this can be through antibody therapies. Antibodies can also be used as a delivery mechanism as well. We heard one of the companies on the CMT RF pipeline looking to use this, as well as immune cell modulation. And overall, it's like, why does it matter that more companies are investing in CMT? And it's been highlighted before. You can't hit the target if you've never taken the shot. We learn, we readjust, and we take another shot, and we get closer and closer to the mark. And this is something that I really think that patients should understand. It's not a wasted effort if you participate in a clinical trial and it's not super positive results. It's the whole field being able to readjust, take another shot, so that we're more on target next time round. So, from patient perspective, I would say that the uncertainty still remains, but the path is clear. And I want all the patients to know that this is a community effort, bringing a therapy to reach patients actually, and they have a role to play themselves, whether they're part of the patient organizations, whether they're funding, whether participating in weird natural history studies or the clinical trials themselves. This is a community effort, and it takes a community to bring a drug to patients. And with that, thank you very much. Any questions? Great. Thank you.
All right, thank you, Rob.
Sorry.
Uh,
I was just gonna ask while you're walking back up there, is there a particular like past therapy that you feel like we've learned a particularly valuable lesson from, or one that's maybe in development right now that gives you a lot of hope? What I guess is what keeps you going every day and what keeps you wanting to keep pushing forward with these therapies and development?
So I should say I'm a basic researcher. So I'm in the lab playing around with stem cells and mouse models and trying to understand a bit more. I'm not really, if I can translate a therapy, I do it more often, but yeah, it's a bit difficult. I think yeah, I know, but I think the field, like, I think Mary answered this really well, that the field really learned a lot from the ascorbic acid trial and the Farnex trial. And it's like I said, you're readjusting, you're understanding what's sensitive, what's not, how can you move forward from this, and you're always assessing what have you learned and where can you go from here. So I think the earlier clinical studies helped the whole field understand that we need better, more sensitive to move forward for therapy. Yeah. Okay.
Thanks, Rob. Actually, it's not really a question, more a comment. I think in light of what you just said, lessons learned is so important. And for those lessons to be well learned, I think all the negative results that come up, both in a lab situation and from drug companies, need to be out there so people can learn better as to why something failed. I think negative results are as important as all those positive results that come in out in CNN or wherever, or BBC or whatever.
Yeah, I think transparency is very key in terms whether you're a biotech company, whether you're a basic researcher. Transparency in how something is done and how people can do the same or get the same result is key. And I know from being a basic researcher myself, when I'm reading someone's method, I can tell whether or not it's going to work straight away. It's like whether this is clear or not. And I'm sure that there's maybe we should have more of a communal reporting, whether it's if it's for basic research or for clinical trials. Normally, they should be registered on clinicaltrials.gov, and you can have a bit of an idea about how the assessment was. But I wouldn't be best to answer for the clinical trials.
Just to add to that, I mean, for example, this was almost, I think, a decade back when I think it was Biogen who ran a clinical trial with dexpramipexole, which in itself was off-the-shelf drug for ALS. And the, the, I think it was phase 3 that failed, but then the results showed that uh, there was a reduction in eosinophil count. And so that led to it being used for a completely different indication that was not known. So there is help with
Yeah.
failed clinical trials.
Okay.
Thank you very much.